presentation by Professor David Steigmann from Berkeley University of California, United States.
Thank you very much for the very kind invitation. And thank you also for accommodating the large time difference between St. Petersburg and California. The problem I would like to describe here is the mechanical behavior of lipid bilayers, so-called lipid bilayers. These are some former students and collaborators that I wish to acknowledge. A brief summary of the topics I will discuss. And for motivation, the motivation comes from biophysics and biology. The lipid bilayer is essentially the membrane that encloses an animal cell, for example, typical animal cell, and which constitutes its interior structures, for example, the envelope of the nucleus, and various structures, for example, the endoplasmic reticulum in the interior of the cell, which adjoins the nucleus. This is an interesting interplay between physics and geometry. For example, here on the right, these small black dots we identify as transmembrane proteins embedded in the bilayer surface. And these are responsible for a large number of cellular functions. They diffuse in the surface of the bilayer, and they mediate shape. They couple, the diffusion is coupled with the shape evolution of the bilayer. So there's an interesting coupled mechanical and diffusion process going on. The basic structure of the bilayer is here at the bottom. It's essentially a liquid crystal, a very thin film of liquid crystal, and consisting of two sheets or two leaflets composed of oriented molecules, polar molecules. The blue here are the so-called head groups for the molecule, and the tails here are the hydrophobic tail groups. The head groups are hydrophilic. They have an affinity for the surrounding aqueous solution, or water. The hydrophobic tail groups will do whatever it takes to shield themselves from the aqueous solution. And here we have a self-assembly mechanism in which the tail groups are adjoining each other so as to exclude the surrounding aqueous solution. And this very simple mechanism gives rise to an enormous range of interesting physics and mechanics as well. So this provides some motivation for the study. Also, the basic molecular structure here is similar to that of a surfactant that one would have at the interface between immiscible liquids. And so there are applications also in chemical engineering that are very interesting. So essentially, the lipid bilayer is a thin liquid crystal film. If we try to bend the surface, so imagine this interface between the two leaflets of the bilayer as a mathematical surface. If we try to bend the surface, then we cause a splay in the liquid crystals, and this is energetically costly. And so in effect, the surface has a bending resistance like a shell, except that the shell has a liquid-like property. So I would like to describe the deformations of that interface, and we'll adopt a standard differential geometric approach in which we parameterize the surface with position using two Gauss parameters, θ1, θ2. By differentiation, this induces a tangent basis in the surface, an associated metric, dual basis, reciprocal basis, and an orientation of the surface, as well as a curvature of the surface, and the B sub alpha beta are the coefficients of the second fundamental form in the surface. So we will apply standard differential geometry to the description of the problem. The curvature is because of the flexural stiffness of the bilayer. The curvature plays a significant role in the mechanics. The main parameters of interest are the so-called mean curvature, h, which is a half the trace of the curvature tensor, and also the Gaussian curvature, the determinant of the curvature tensor. And these are expressed here in component form. As I mentioned, the bilayer behaves mechanically just as a classical Kirchhoff-Love shell, essentially. And so if one is interested in equilibrium, one has an equilibrium condition of this form. For example, suppose there is an osmotic pressure acting laterally on the surface, which is typical in the biophysical context. Then this is balanced by a divergence of a stress-like quantity, given explicitly here, where small a is the determinant of the metric in the surface, positive definite. And the stress vectors, T alpha going from 1 to 2, can be decomposed into a tangential parts here, N alpha, and a transverse part, S alpha, which is associated essentially with transverse shear tractions acting on the surface. And these arise from bending effects. So they're balanced by essentially a divergence in the surface of a bending stress. And the tangential components involve a conventional stress, which is coupled with the curvature to a bending stress. This conventional stress is, as in conventional elasticity, related to the derivative of, say, free energy per unit mass with respect to the metric in the surface, a symmetric tensor, analogous to the Cauchy stress of continuum mechanics. And because of the possible dependence of the free energy on curvature, there are also the bending stresses. So this is, the lower half of the page here is the standard, a synopsis of the standard Kirchhoff-Love nonlinear shell theory. What is interesting about our shell here is that it's essentially a liquid shell. One can show either by a dimension reduction procedure from three-dimensional liquid crystal theory, or perhaps more appropriately from material symmetry arguments for the actual shell itself regarded as a two-dimensional continuum. One can show that the dependence of the energy, I'll call it F here, on the curvature is via the mean and Gaussian curvatures, and also possibly a density, a mass per unit area in the surface. The phenomenology indicates that it's energetically costly to dilate the surface or to change its local surface area in comparison with bending of the surface. And so we introduce a Lagrange multiplier field to suppress density changes in the surface or areal deformations. So essentially, this is a two-dimensional analog of incompressibility, an area incompressibility. Also, the bilayer, if we, back to the figure here, is unoriented. That is, there's a reflection symmetry of the system with respect to the interfacial plane. That reflection symmetry implies that the energy is an even function of the mean curvature. K, the Gaussian curvature, is already a quadratic function of the curvature and so is unaffected by this argument. Typically, it's more advantageous to introduce an energy per unit area. And because of the area constraint, that can be either per unit current area of the actual configuration, or per unit area of a reference surface, a fixed reference surface that we might use as a computational domain, for example. If we project these equilibrium equations into the direction of the normal to the surface, the interface, we can calculate the pressure, and that works out to be this fairly complicated extension of the classical Laplace equation of capillarity, except here extended to accommodate the bending resistance of this liquid crystal film. W here is the bending energy. If all terms involving W are suppressed, then we recover the classical capillarity equation, that the mean curvature is balanced by the pressure. And lambda is effectively this Lagrange multiplier, a combination of the Lagrange multiplier with the energy that's constitutively indeterminate, must be determined simply from the boundary value problem that we wish to solve. Here, the subscripts H and K refer to partial derivatives of the energy with respect to these variables. This is the two-dimensional Laplacian in the surface, a symmetric second covariant derivative. B tilde here is an interesting geometric quantity. It's the cofactor of the curvature tensor. And it has the useful property that its divergence vanishes identically. And that's simply a restatement of the classical Minardi-Codazzi compatibility conditions in the surface. And so we have a rather complicated replacement of the classical Laplace equation of capillarity, which involves the differential operators in the surface. If we now project these equilibrium equations into the tangent plane of the surface, we obtain ultimately simply an expression that controls the gradient of the Laplace, the Lagrange multiplier in the surface. And that turns out to be balanced by the explicit derivative of the energy on coordinates in the case of a non-uniform film. Later in the talk, I will discuss possible sources of that non-uniformity. So for a uniform film, the right-hand side would be 0, lambda would be uniform in the surface, and then with the suppression of the bending terms, we'd recover exactly the classical Laplace capillarity equation. Well, topics in this of interest in this subject include wetting of the substrate, for example, and associated boundary conditions. One can proceed instead of following the balance laws for a shell. One can proceed, of course, from a variational argument. The Euler-Lagrange equations are exactly as those we have stated a moment ago. When they are satisfied in the body, in the interior of a patch of the surface, then there is a residual virtual power expression associated with boundary working, and that can be used to generate, of course, boundary conditions in the usual manner. And that breaks up into two parts, say a tangential boundary working of this form and a boundary working in the normal direction, normal to the interface. We can combine these together, add them together, and with some rearrangement, we can cast these in a more conventional form. This vector in parenthesis here is essentially a traction vector in the boundary. U is a virtual velocity, and this traction can be decomposed into a part normal to the boundary but lying in the tangent plane to the surface, a part tangential to the boundary, and a part normal to the surface, normal to the interface at the boundary. And these are associated with in-plane tractions, a normal traction, a shear traction on the boundary, and a transverse shear traction in the direction of the normal, as well as a bending moment that is power conjugate to a rotational virtual velocity. If the patch of surface that we're studying has corners, places where the tangent to the boundary is discontinuous, then we also have so-called corner forces at those corners, which are found in this way. And this format is identical to the classical Kirchhoff theory of plates. You may recall from your school days the notion of corner forces in that context, and we have them again here. To describe the wetting of a liquid bounded, say, by a bilayer with a substrate, the conventional phenomenological approach is to append an adhesion energy to the system. So the sigma is an adhesion energy per unit area, and this area here is the area of the wetted part of the substrate. Here we will, if the substrate is uniform, then the adhesion energy per unit area will be uniform as well. The positive value of sigma promotes wetting. It lowers the energy by increasing the wetted energy. Negative sigma discourages wetting of the substrate. So here I will consider a non-uniform substrate in which sigma may depend on position on the substrate, and that what we need to secure boundary conditions is an expression for the variational derivative of this adhesion energy. And there's some complicated considerations here, which I won't discuss in detail. But the final result of which is that the variational derivative of that adhesion energy can ultimately be written as a boundary integral of this form. Again, U is the virtual velocity. This vector T, if I can move forward, is tangential to both the bilayer here. There's a bilayer and a bulk liquid, water for example, rigid substrate. T is tangential to the substrate and is normal to the bilayer at the contact line. Capital N is normal to the substrate. Nu is normal to the bilayer in the tangent plane of the bilayer. Okay, so The classical Young-Laplace equation is an equation of equilibrium that tells us the contact angle that's formed between the film and the substrate, and we're interested in generalizing that condition. So here we have the variation of the adhesion energy that can be now incorporated into the expression for the residual boundary working, or the residual virtual work statement, a virtual power statement in the boundary. From the point of view of liquid crystal theory, the interesting boundary condition is associated with so-called conical anchoring, where the angle gamma between the normal to the bilayer and the substrate is prescribed on the substrate. And it could be a function of position on the substrate if it is a non-uniform, if the substrate has non-uniform properties, for example. And so we are importing essentially a typical condition from liquid crystal theory into this topic. With some analysis, then, we can extract the generalized Young-Laplace equation that allows for a variation in the so-called cone angle. So this angle allows the normal to the substrate to form a cone relative to the normal to the interface to form essentially a cone relative to the normal to the substrate. This is the reason for the name conical anchoring condition. And we have then a generalization of the classical Young-Laplace equation. And we have also an additional non-standard equation, which arises solely due to flexural stiffness and is therefore absent in the classical capillarity theory. And in the absence of flexural energy, this reduces precisely to the classical Young-Laplace equation involving a surface tension, cosine of the contact angle equals the adhesion energy. And these intervening terms are associated with bending in the substrate, bending energy, sorry, of the bilayer. Well, to study some simple solutions, we can imagine linearizing the equation for the shape with respect to a plane. And so it's natural then to adopt the Monge parameterization of the surface. Theta is position on the plane. Z of theta is the height of the bilayer above the plane. And we can contemplate linearizing the theory for small z and small gradient of z. And some calculations here lead to that, ultimately to that linearization. And here it is at the bottom. K is a bending modulus in the energy. The subscript P refers to a Laplacian on the plane, not on the film itself. And lambda is the Lagrange multiplier we studied before. This is simply the classical plate bending equation, elementary plate bending equation, with an in-plane surface tension or compression lambda. And it's a simple matter to solve that. We can recast that using the fact that in the linear approximation, the mean curvature is 1/2 the Laplacian of z. We can recast that equation as this Helmholtz equation. And I'll take the Lagrange multiplier to be positive and call it mu squared in this context. And we'll take it to be an assigned positive constant. And of course, Laplacian z then is proportional to the mean curvature according to this equation that's also proportional to the Laplacian of the mean curvature. So we collect everything under a single Laplacian. The square bracket then can be any plane harmonic function phi. And so we have the general solution to this linearized problem. Here is the Helmholtz equation in polar coordinates. Its general solutions involve Bessel functions of the modified Bessel functions of the first and second kind, that is Bessel functions with complex argument, as well as the trigonometric functions. If we want to solve an exterior problem, exterior to a circle, for example, in the polar system, then we seek a bounded solution, let's say a far field, a bounded solution bounded in the far field. And so the only terms that survive are the modified Bessel functions of the second kind. Our plane harmonic function has solutions of the standard form, shown here. A is the radius of a boundary, and R is exterior to that, is greater than A. And so we can compose the far field solution, or the solution to the exterior problem, valid if, in the case of asymptotic flatness, if far away from the origin, if the bilayer approaches a plane, then this is the general solution. So here we will apply this to a simple problem. Let us suppose we have a substrate in the form of a cylinder. Here is the bilayer, and there's an aqueous solution below the bilayer, water. And we will suppose that the substrate is hydrophobic, so that the adhesion energy, wetting energy, is negative. So the effect is to repel the water phase, and this induces a bending in the bilayer. We will prescribe this conical anchoring angle as pi/2. That is, this is justified by the idea that the hydrophobic tail groups in the bilayer would then be shielded from the aqueous phase by the hydrophobic cylinder to which it's attached. The effect is, in the boundary conditions, is to induce a transverse shear force on the bilayer, which is exactly this wetting energy. And the larger the magnitude of the adhesion energy, then the larger is this transverse shear force, which has the effect of pushing the bilayer vertically downward. Also, the transverse shear force is related constitutively to the normal derivative of the mean curvature in the membrane at the boundary, and K is a bending modulus associated with a specific form of the energy. And for an axisymmetric problem, the solution simplifies dramatically to this form. And we can compare it, the linear solution I've just shown you is in the dashed line here. We can compare it to the nonlinear solution obtained numerically for larger values of sigma, which is effectively associated with increasing values of the transverse shear force. So for larger sigma, you expect a deeper penetration, and therefore a greater deviation of the nonlinear solution from the linear one, and that is in fact shown. That is in fact predicted. And interestingly, the linear theory underestimates the deflection relative to the far field value. The linear theory is overly stiff, in other words. Another interesting problem in the subject is adhesion of a closed cell, a closed vesicle with a substrate. So we again have an adhesion energy here. There's no boundary of the film, but there is essentially a free boundary, the perimeter of the contact region of the bilayer with the rigid substrate. So I will call that omega sub C for contact. The remainder of the bilayer I'll call omega sub F for freestanding portion of the bilayer. And the perimeter is essentially the transition from the contacting to the non-contacting portion of the bilayer. And we must determine this contact region. We can use our residual virtual power statement by dividing the bilayer into two parts and regarding this perimeter as the common boundary of the two parts. When we combine the two virtual work statements into one, then we find that there's a potential for certain fields to be discontinuous at the perimeter. And we indeed find that that's the case. So here's some analysis associated with the reduction. The equilibrium conditions at the perimeter are simply these here. The square bracket is associated with the jump, the discontinuity, as one approaches the perimeter from the freestanding portion of the bilayer, subtracts the field coming from the contacting portion. We find essentially two conditions that must be satisfied involving discontinuities. And ultimately we can show that the first of them reduces to an identity, but the second one yields non-trivial information. And this involves the discontinuity in the curvature of the bilayer, which is necessary to sustain adhesion. So that's an interesting aspect of this problem. although the tangent plane to the bilayer is continuous, the curvature at the perimeter is actually discontinuous. If we adopt a specific energy of the classical type known as the Helfrich energy, which is quadratic in the curvatures, then with an associated bending modulus K, then this condition reduces simply to this one. The square of the discontinuity in the mean curvature is balanced by the adhesion energy normalized by the bending modulus. And that shows that, of course, adhesion would be impossible if the adhesion energy were negative, right? Which is intuitively plausible. We can also apply loads, for example, a point load at the pole of the membrane to either promote adhesion or to detach the cell from the substrate. That point load is easily obtained by an equilibrium condition. For example, consider a subsurface omega tilde containing the pole where the point load is applied. We can use a simple force balance. This is the traction distribution on a parallel of latitude and the pressure acting on that subsurface. We take the limit as the diameter, the radius of this subsurface tends to 0. We obtain the point load in terms of a limiting value of the transverse shear force, and that can be used to integrate the equations, effectively provide a boundary condition to integrate the equations. Normally in an axisymmetric problem, we would use arc length as the independent variable. However, because we are prescribing area, we have a constraint on area, it's more convenient to use the area associated with a given value of arc length. So we can substitute in place of arc length this area associated with arc length S, and that's possible because there's a one-to-one correspondence between this area measure and arc length itself. So the convenient way to proceed is to integrate the equations for this area variable between 0 and a prescribed total area, and rather than use arc length. So that's the procedure we will use. And so we solve the differential equations augmented by this continuity condition at the perimeter. What we do in practice is to prescribe a contact area and then use the theory to calculate the point force that is associated with that contact area. Normally one would prescribe the force and compute the contact area, but numerically it's easier, more straightforward to do the reverse. So here's a calculation. For example, we prescribe the contact area. We use the theory to compute the point force associated with that contact area. So one can prescribe the sequence of contact areas, as we've done here, use the theory to compute what the point load must be associated with that contact area, and compute numerically a force contact area relationship. So as you would expect, for large negative point load, there's a large contact area. The bilayer is being pressed into the substrate. As we diminish the intensity of the load, the cell deflects upward. This is the configuration associated with 0 load. So the only external influence on the bilayer is the adhesion, which has the effect of compressing an initially spherical bilayer, for example, into the substrate. And then as we transition to positive point forces, we have a deformation like so. What we compute is that there is a maximum to the point load associated with any area. So naturally, as we pull up on the bilayer, the contact area decreases. That there's a maximum load in equilibrium. So if we attempt to impose a larger load than this maximum, there will be no equilibrium state, and there will be a dynamic detachment from the substrate at a finite value of contact area. And that, in fact, is experimentally observed. So that's a satisfying result. We can change the shape of the substrate if we wish using the theory, and we have the same sort of qualitative behavior. Again, the same qualitative behavior. Let me pass over this problem. If we'd like to introduce some physics into the problem, so far we've discussed pure elasticity as in an elastic shell. For example, we can revisit our problem of proteins diffusing in the endoplasmic reticulum of the bilayer of the cell. So for example, suppose we have a concentration of proteins in the bilayer, and I'll suppose the number density of proteins is sufficiently high to justify a continuum approximation. This complicated slide here is a discussion of the distinction between convected coordinates that we can use to label lipid molecules, for example, the distinction between those and coordinates that are fixed in the surface. So this is entirely analogous to the Lagrangian versus Eulerian description in conventional continuum mechanics. And for example, a dot here is the material derivative, that is a time derivative following the lipid molecules. The subscript T pertains to a time derivative in the surface holding the surface fixed coordinates constant, and then we have a convective term. So this is analogous to the standard continuum mechanics. But this formalism facilitates the derivation of a transport equation, which is useful in, for example, in studying the diffusion of proteins. So we'll suppose that sigma here is not the wetting energy, but I'll use the same notation to denote an areal density of proteins, a number density of proteins, number of proteins per unit area of surface. And for example, then the rate of change of the number of proteins in a patch of the surface will be balanced by a diffusive flux vector through the boundary of the surface. I could add a source term coming from the bulk, but that's an easy thing to do, and so I suppress it here. And using the convected coordinate formalism and the associated transport equation, we can derive a local balance equation, which involves this tangential diffusive flux vector in the form of a divergence on the surface. This combination here is associated with the areal dilation of the surface. J dot is the areal dilation, areal stretch. J dot over J is simply this parenthetical term. This is the divergence of the tangential velocity field balanced by the mean curvature times the normal velocity in the surface. And the remainder are the standard terms. If the concentration is sufficiently dilute, then we would not expect it to disturb the areal incompressibility of the bilayer. So it would be a good approximation to retain the constraint of no energy change. And then the diffusive balance simplifies accordingly. Okay. And then we have the normal velocity coupled to the divergence of the tangential velocity field. Okay, well, I want to account for the fact that diffusion is, of course, a dissipative process. We define the dissipation to be the difference between the power supply to a patch of the membrane, protein composite, and the rate of change of the energy contained in the membrane protein composite. Now we will allow the energy per unit area in the film to depend also on protein concentration, in addition to the standard variables that we discussed before. The power supply is the usual mechanical power supply, forces, pressure, bending moment at an edge. We also have an energy supply rate due to the diffusing proteins, Q. It's analogous to a heat flux vector. It's given by the chemical potential, the derivative of energy with respect to protein concentration, times the diffusive flux vector M. And as a surrogate for the second law of thermodynamics, will demand that the dissipation should never be negative for any patch of the surface. A simple proposal for the energy, explicit form of the energy would be this. This is quadratic in the curvatures, and that is justified by the idea that if we non-dimensionalize the curvature by the thickness of the bilayer, then in practice the non-dimensional curvature of the film will be very small, even though its deformations can be large. And so a quadratic approximation is normally sufficient, and that same reasoning applies also in three-dimensional liquid crystal theory. The coefficients can now depend on density or aerodilation as well as protein concentration. A special case of this would be this form here, where C is the so-called spontaneous curvature. This is the curvature at which the energy would be minimized, so we could have a relaxed state of the energy when H, the mean curvature, is equal to the so-called spontaneous curvature. If we allow the spontaneous curvature, conventionally this is a fixed parameter, but if we allow it to depend on protein concentration, then we have an evolution of shape as the system tries to minimize energy, evolution of shape in response to the distribution of the diffusing proteins, which couples the diffusion to the shape evolution of the bilayer. Okay, so we can invoke the balance laws here. The virtual power principle, which simply states the statement of mechanical equilibrium. I'll suppose inertia is negligible. Here, the material derivative of following the lipids of the energy involves now the evolution of the concentration of proteins. And combining these, we have an expression for the dissipation then, which can be reduced using the diffusive balance law to this simple form. And so we have effectively a constitutive inequality analogous to what one finds in heat transfer, which is that the diffusive flux is opposed to the gradient of the chemical potential in the surface. And so we would adopt a simple constitutive equation that is such as to satisfy this identically. For example, the classical situation, the diffusive flux, is a negative constant. That is to say, C is a positive number, the diffusivity, multiplying the gradient of the chemical potential in the surface. And if we have a simple form of the chemical potential involving in particular the spontaneous curvature, then we extract the constitutive equation for the diffusive flux, which couples concentration gradient to the geometry of the film. The equilibrium equations are modified slightly as well. And in particular, the gradient of the Lagrange multiplier is balanced by the gradient of the concentration. To choose the form of the spontaneous curvature, this is a simple proposal for a dilute concentration of proteins. Phi here is associated with the geometry of a transmembrane protein. We can think of it as in the shape of a cone or an ice cream cone piercing the membrane transversely. The interactions of that conical protein with the membrane induce a natural curvature. So phi is essentially the cone angle of that ice cream cone, if you can imagine it. And so this is the system we have. And we'll solve some axisymmetric problems for illustration. Here's a simple problem which we have a disk, a circular disk, flat disk, and at time 0, let's say, we expose the edge to a protein bath. So we can here normally we prescribe chemical potential. I think in this simulation we're prescribing actual protein concentration. The proteins then diffuse in over time, and induce, because of their own natural shape, they induce a bending of the bilayer over time. And the distribution of proteins becomes increasingly uniform over time as they coat a shape that's approaching a spherical surface. Here is another problem associated with the geometry of the so-called nuclear pore, a pore in the nucleus of a cell, which is an opening essentially into the interior of the nucleus. We can model it locally, a pore as a kind of a piece of a toroidal surface. And here is an equator. And imagine revolving this curve here about an axis displaced slightly here from this z-axis. We prescribe a flux of proteins here. And over time, then, this induces a bending of the surface. Asymptotically, it becomes this toroidal, flat in the far field. And the distribution of proteins becomes more and more concentrated near the throat of this pore. And that, in fact, is consistent with the observed phenomenology, protein distribution in the vicinity of a nuclear pore. As a final topic in this tour of various topics, I would like to discuss viscous flow in the surface. If you remember, in the beginning, we had a conventional stress, sigma, that I referred to. This is like a membrane stress. In the case of fluid film with a constraint on area, this is simply a conventional surface tension kind of term, or surface pressure term, to which we would add a viscous stress if we want to account for viscosity. And a simple framework would be to adopt a surface analog of the Navier-Stokes theory, in which the viscous stress is a shear viscosity. We're suppressing areal dilation, so we don't have a bulk or a viscosity in the surface. We only have a shear viscosity as a linear function of the straining in the surface, which is essentially the Lie derivative of the evolving metric in the surface. And that Lie derivative is simply the material derivative of the evolving metric. To compute it, we simply take a conventional time derivative in the convected coordinate description of the kinematics. That Lie derivative turns out to be related to the symmetric part of the velocity, the tangential velocity gradient in the surface, and to the normal velocity coupled to the curvature. And so we add this viscous stress, and we can revisit then the balance laws that we had at the beginning. They will involve the divergence of that viscous stress as well. And this D here is simply the symmetric part of the tangential velocity gradient. We can re-examine the balance laws we had before. Here is the areal incompressibility constraint. We've changed the notation slightly. V alpha, the tangential velocity. W is the normal velocity. The gradient of the Lagrange multiplier now is balanced by viscous terms, which involve the shape of the surface as it evolves. This is kind of like a Marangoni flow effect, except modified to account for curvature in the surface. The shape equation balancing the pressure is again modified by viscosity as well. And we can extract boundary conditions. The bending moment is unaffected by the viscosity. The transverse shear force is unaffected, but the in-plane tractions in the normal and tangential directions to an edge now have viscous contributions, as shown here. A simple problem to illustrate would be, for example, this one. Let's study the evolution of a square film. We will apply a pressure transverse perpendicular to your screen to cause an out-of-plane deformation of the bilayer. We'll have impenetrable walls here, 3 of them, where we clamp the bilayer, so it has 0 slope as it deforms, and also no velocity, so we'll have no slip boundary conditions here. On this remaining edge here, we'll prescribe traction, which has the effect of facilitating a flow of lipids into the computational domain from the exterior. And we will prescribe a lateral pressure as a function of time and study the evolution of the dynamics. So we have a coupling between the evolving shape of the membrane and the flow field in the surface of the bilayer as well in response to the pressure evolution. And we can see an interesting coupling that generates vorticity patterns in the surface. These are the streamlines of the velocity field. One imagines that this is perhaps not so interesting in the biophysical context, but perhaps more interesting in the chemical engineering context, in which this would be a surfactant film at the interface between immiscible liquids. And that's the end. Thank you very much. Spasiba.
Thank you very much, Professor Steigmann, for a lecture. And now, please, questions.
I can supply papers to you if anyone is interested in some details that I've skipped over lightly.
Yes, of course. Yes, and here we have a question from Evgeny Aborin. To the force deflection responses, there are different contact areas for a single value of the lumped force. What does this ambiguity mean? And what methods do you use for solving the BP?
Well, the simulation I just showed you is conducted using a program called COMSOL, which is essentially a commercial finite element package for multi-physics problems. What was the other part of the question? I'm sorry, I didn't follow. The beginning of the question, can you repeat it, please?
What methods do you use for solving the BBS?
The BBS, what is the shape equation? Or, so in the axisymmetric problems, we use a standard ordinary differential equation solver like MATLAB or a shooting method, Euler backward and so on, or Runge-Kutta method. In the more interesting non-symmetric problems, we use a finite element in the surface.
Okay, thanks. And can I ask techniques to switch on microphone to Victor Yeremeyev?
All right.
Okay, let's switch on.
Yes.
I ask.
Victor, can you ask a question, please?
Okay, dear David, first of all, thank you for a nice presentation.
Can you speak closer to a microphone, please?
Yes.
Okay, I'm trying to speak louder.
Okay.
Simply louder. First, I have several questions because it's so many topics, but I'm
yes, too many. I apologize for.
No. I'm just for fluids. Is it possible also to add rate of curvature, because it looks nice? Your energy, static energy, depends on two tensors, so it could be natural to consider it also the rate of curvature.
Yeah, of course, one can have, in other words, a kind of viscosity with respect to curvature, not just metric, right?
And second question, unlike to classic capillarity, I guess here some unbounded solutions are impossible. It's true?
Unbounded solutions? Well, so the presence of bending stiffness, for example, would suppress unbounded curvature, which one could find in a classical capillarity at an edge, for example. Here in this theory would be energetically impossible. It would require unbounded energy. So
even for
the bending terms exert control, energetic control over the solutions.
Okay, even for very hydrophilic conditions, you'll also control this.
It depends on the context, I suppose. What problem do you have in mind exactly?
Because in classic capillarity, for example, without gravitation, there are shape of cap, for example, when you can't store a liquid.
Yeah, I would think that the bending energy will
prevent it.
Has a regularizing effect, and one does not.
Okay, thank you very much.
Yeah, the viscous bending terms, it's difficult to find a physical justification for those. It seems as the surface flexes, the interactions between the lipid molecules in that mode of deformation are essentially elastic, but the lipid molecules can flow in the surface as a conventional liquid on a curved surface. And so there seems to be a more substantial physical motivation for considering the rate of change of metric rather than rate of change of curvature in the constitutive functions, at least. But
okay,
the constitutive
I see.
Level. Yeah. And of course, one can study also a genuine viscoelastic fluid, a rheological fluid. This model here I presented was simply the simplest viscous contribution that one can make to the tangential membrane stresses. But one can contemplate a genuine rheological model, as in viscoelasticity.
Good. Thank you so much.
Thank you.
Thank you very much. Maybe does anybody have any more questions? No.
I see Julius Kaplanov on the screen, so I'd like to simply say hello, Julius.
Hello, David. Then I should ask a question. Well, I'm not asking with this advanced technology, but still, it might be a potential to explain better the related bending stiffness, as you mentioned, just without energy, but through equilibrium, variation through thickness, whatever, in a very old-fashioned manner. Maybe physics may come from there. What do you think?
Well, yes. One can extract the shell model, which is a two-dimensional continuum model. One can derive it from three-dimensional liquid crystal theory via an asymptotic expansion, as has been done historically by shell theorists in mechanics, right? Asymptotic derivation of shell.
Sure.
That's a drawback in this context is that the shell is really only 2 molecules thick, so we don't really have a three-dimensional continuum.
And it means that you need energy. Yeah, basically.
The theory, the structure of the three-dimensional continuum liquid crystal theory at least gives us guidance as to the nature of a two-dimensional theory for a thin film of molecular thickness.
I see.
Or one can simply use symmetry arguments appropriate to fluidity. For example, adapting ideas of Walter Knoll in the context of continuum mechanics to distinguish between solids and fluids by the use of material symmetry. I skipped over this much too lightly, but there are basically two approaches where one does not need to come from a three-dimensional continuum theory. One can propose a direct model, but I think that the asymptotic derivation from 3 dimensions is actually very instructive and illuminating, and I personally prefer that approach because it offers concrete guidance for the structure of the two-dimensional theory. But yes, classically one integrates through the thickness and three-dimensional continuum theory. But in modern times, I think we use asymptotic derivation methods, or even methods based on so-called gamma convergence, which is a rigorous dimensional reduction.
Yeah, for instance. Yeah, thank you, David.
Thank you. Good to see you.
Good to see you. Take care. Bye.
Good to see you.
Yes, Professor Steigmann, thank you very much for your lecture.